Hi, this is Yaya and welcome to another video for MCDB 427, which is molecular biology at the University of Michigan. The figure that we will be talking about in this video is figure 17.9 from the 5th edition of Molecular Biology by Robert F. Weaver. The purpose of this video is to present to you and walk you through the experiments that were done in order to get a better understanding of IF2, its role as a GTPase, and how it helps initiate translation in prokaryotes. However, before we start talking about this, I want to say that the 5th edition is wrong in referencing the correct paper for figure 17.9. The information for the paper from which this data comes from is below. The best way to go about exploring the role of IF2 as a GTPase is to start by explaining the basics of translation initiation in prokaryotes as we know it now. This cartoon shows us an mRNA fragment interacting with the 30S pre-initiation complex, which includes the 30S ribosomal unit, the initiation factors IF1, IF2, and IF3. Notice that IF2 is bound to a GTP. The cartoon also shows us that within the 30S pre-initiation complex, the FMET tRNA FMET is present. In fact, the IF2 GTP amino acyl tRNA arrive as a complex. The next thing that happens is that the 50S subunit, which is shown over here, comes in and joins the pre-initiation complex and makes it so that the initiation factors leave. Therefore, the question that the researchers were trying to address was whether GTP hydrolysis plays a role in this process and whether or not GTP hydrolysis even takes place. Now, before we can address questions, we should cover how the researchers actually knew that it was indeed IF2 that interacted with the GTP and not IF3 or IF1. And to figure this out, in the same paper, the authors conducted a binding assay using radiolabeled GTP and ribosomes with or without the initiation factors. As you can see, when they did the binding assay with ribosomes alone, they noticed a minimal amount of GTP binding. They then decided to see what would happen with GTP binding if they chucked in IF1 with the ribosomes, and there was no difference. They decided to do the same thing, but this time they used IF3, and again, there was no difference. But when they did the binding assay with ribosomes and IF2 in the presence of GTP, there was a huge increase in the GTP binding. Also, if you take a look at the amount of IF2 they used, you can see that this is a lot less than the amount of IF1 and IF3 that they used in their experiments. And again, there was a huge increase in the GTP binding with just this small amount of IF2. This led them to conclude that it was indeed IF2 that interacted with GTP. Now that they knew IF2 brought in GTP to the ribosome and was the GTP binding protein, they could work on the fundamental question of whether or not GTP hydrolysis took place. To go about doing this, the experiment that they set up was fairly simple. The authors mixed 32-phosphate gamma-labeled GTP with either salt-washed ribosomes, IF2, or both, and decided to plot the release of the gamma-labeled inorganic phosphate if hydrolysis occurred. The reason why they use salt-washed ribosomes is because ribosomes usually have many proteins and factors associated with them, and salt wash helps get rid of these. This is the figure of the plot as shown in the actual paper. Because this paper was published a while ago, and the figure itself is a little blurry, I took the liberty of writing down what the x-axis and the y-axis say. So the y-axis shows the amount of GTP hydrolysis, and the x-axis shows the time. So if we were to start breaking down this plot, we would see that at the bottom of the graph, there is a line that corresponds with IF2 in the presence of gamma-labeled GTP. And as you can see, there is really no hydrolysis going on there. The line above shows GTP in the presence of just ribosomes. And though there's a slight increase, this can be due to contaminations and a poor salt wash. But finally, at the top, we have this line over here, which shows a drastic increase in the hydrolysis of GTP. And this hydrolysis happens when they used IF2 in the presence of ribosomes. This led the researchers to conclude that IF2 is a ribosome-dependent GTPase. Now that we have had the data which allowed the authors to conclude that GTP hydrolysis is indeed involved in translation initiation of prokaryotes and that IF2 is a ribosome-dependent GTPase, let's put this information within the context of the actual molecular mechanisms of initiation. Figure 17.11 from the book shows a summary of this. In this figure, we can see the 30S ribosomal subunit, which is shown here in blue, interacting with all three of the initiation factors. Note that IF2 is bound to the GTP. It is also interacting with the FMET tRNA and an mRNA fragment to form the 30S pre-initiation complex. This is followed by the addition of the 50S ribosomal subunit, which is shown here in pink. The 50S ribosomal subunit kicks off initiation factors 1 and 3. After this, the IF2 bound GTP is hydrolyzed and kicked off, allowing for the formation of the 70S complex, which can then initiate translation. Thank you for watching this video. I hope it was helpful, and as always, go blue.